All right. Hi, everyone. I hope that you are all doing well. You should have gotten an email from me right before I came up here with the info about the review session. So based on the doodle poll, the most popular time was on Sunday at 7. So that's when it will be. It will be on Zoom. The link is in the email. I am going to record it, but I would say don't just say, oh, well, I'll just watch the recording later, because if no one actually comes and asks any questions, then there isn't a review session. So there's nothing to record. Just be aware of that. There is a whole bunch of practice material on Moodle, and you guys are going to get an email from me with your graded VDJ problem sets, as well as other things coming soon. As I spend my weekend reined in, because it's going to be a hurricane all weekend, so I assume you guys might have all the rain this weekend too. So today we're going to talk about B-cell development and selection, and sort of finish going through the process of how we develop B-cells. So if you recall, we're sort of thinking about these yellow steps of what is happening in the life of the B-cell. In fact, really, we largely talked about this idea of generating diverse B-cell receptors in the bone marrow. Today we'll see some of the selection pieces of this. And of course, this is all happening in the bone marrow, both as I said when I read that box, and as you can tell from the color coding on this slide. And last time we saw the pro-B cell and the pre-B cell, and we thought about some of their sort of important criteria. So with the pro-B cell, that pro-B cell was doing D heavy to J heavy rearrangement at an early stage, and then V heavy to DJ heavy at a later stage. And it had no BCR on its surface. We also had the pre-B cell. The pre-B cell was first testing the heavy chain, and then it was doing light chain rearrangement, and it had the pre-BCR on its surface, which is the heavy chain plus surrogate light chain. And the name of surrogate light chain is lambda 5 or V pre-B. Actually, both of those together are the surrogate light chain. And we got to the immature B cell, which has an actual BCR on its surface once we have finished making our light chain. So here you can see some of those sort of earlier steps that we talked about with the pro-B cell and the pre-B cell. And we talked about some of the things that happen to the pre-B cell when it is getting signaling through its receptor. So some of the parts of testing the heavy chain before we get to making light chain. And if you recall, we talked about a big part of testing the heavy chain as being the situation where we have both chromosomes doing the D to J rearrangement of the heavy chain. They both kind of try to do V to DJ, but one of them finishes first, because that's how time works. And if that was a successful protein, this cell is now going to have this protein available to get a signal, which will stop further rearrangement at the heavy chain, as well as do things like give a survival signal, a signal for proliferation, turning off RAG1 and RAG2, and as I said, that allelic exclusion, turning off production of the surrogate light chain. If the cell does not make a good protein here, then it will try to rearrange the other chromosome. There will be no protein to give a signal to say stop, so it will keep going on that other chromosome. If that works, the cell is going to kind of signal as a pre-B cell. If not, the cell is going to die because it really is needing signals through its receptors in order to be told to live. And so once our pre-B cell has tested its heavy chain, it can now go ahead and make a light chain. So it's going to take the V of a light chain and J of a light chain and start doing that rearrangement. If it is successful, it will put the heavy chain and the light chain on the surface, and now it gets to be called an immature B cell because it has this different thing on the surface. This is where I really left you last time, but there are a few kind of additional details we need to think about with the light chain rearrangement and exactly how that light chain is going to be tested. So of course, as you might imagine, one of the things that's going to be happening with the immature B cell is going to be testing the light chain. Though, as you will see, there is another piece to this. But one complication that comes up in doing light chain rearrangement is that there are two places in the genome where there are light chain genes. So we have a type of light chain called the kappa light chain and a type of light chain called the lambda light chain. They differ based on if they have the kappa constant region or the lambda constant region. The kappa is on chromosome 2, lambda is on chromosome 22. They both make light chain. With the heavy chain, we talked about how, IgA versus IgE has different functions. Kappa and lambda, to our knowledge, have no different functions. They're exactly the same. They're both just light chains. But what is unique here is that now the cell has more chances to try to make a light chain because it has both the chromosome containing kappa and the chromosome containing lambda. And when the cell does its rearrangement, it always chooses to try kappa first. We don't know why, but it always does kappa of one chromosome, either mom's or dad's. And if that one works, then woohoo, we're an immature B cell, and we get to do immature B cell stuff. And if that one doesn't work, then we do the other kappa. And if it works, woohoo, we get to be an immature B cell and do immature B cell stuff. And if it doesn't work, then we try lambda. And if lambda works, then woohoo. If not, you use the other lambda. So we've got more chances here to make a light chain. As I said, there is no known difference between a light chain using kappa and a light chain using lambda. The only thing that we know is that if a B cell uses lambda as its light chain, it must have tried and failed at least twice to make a light chain because it must have tried and failed kappa 2 times before it got to lambda. That's really all we know about it. If that cell is successful, then we're going to get that B cell is going to get to go into this selection process that we'll come into. If not, it's going to try again. And so here is sort of that same version of that B cell trying again. So first it does D to J of the heavy chain. It tries 1 V to DJ. Maybe it works, so it gets to go on. Maybe it fails. When it fails, tries a second time. Maybe it works. Maybe it fails, and then the cell dies. After we've gotten a successful heavy chain, now we can try kappa once, and then kappa, the other kappa, and then we can try lambda once, and then we can try the other lambda. And if it still doesn't work after all that, the cell is going to die because it's not able to get signal. When the cell does successfully get a signal through the heavy and light chain together, it will, that signal will again tell the cell to turn off RAG1 and RAG2. So again, we're going to turn off RAG1 and RAG2 at this point. That's important because RAG1 and RAG2 can damage the DNA, and we don't want those risky enzymes on any longer than we need to. But it's also important so that we can make sure that we get allelic exclusion here. Because just like we need to do allelic exclusion of the heavy chain, we also need to do allelic exclusion of the light chain. And so here is that same slide I've shown you before about allelic exclusion. This might work. Oh, yeah, this will work. But let's think for a second what would actually happen if allelic exclusion didn't occur. On that slide, you see an example on the right of sort of a B cell that has not done allelic exclusion at the heavy chain. And one of the examples, the 3 different receptors you see, is not symmetric. It has 2 different heavy chains and 2 different light chains. I'm going to ignore the not symmetric options when I draw this out, just because if I drew those 2, it would become insane. So we're just going to look at symmetric options of what would happen if we didn't have allelic exclusion here. So we could have a heavy chain from mom. We could have a heavy chain from dad. We could do a mix and match, but I'm not drawing those asymmetric ones. And then we could have mom's kappa paired with either of them. We could also have dad's kappa paired with either of those heavy chains. Or we could have mom's lambda paired with either with those heavy chains. Or we could have dad's lambda paired with either of those heavy chains. And so if I'm not even counting the asymmetric ones like this, it's so dramatic that I'm not counting them that I knock things over. If I'm not counting these asymmetric ones, this cell would have 8 different receptors. And that would be really bad for that cell, both because it could not bind to anything very strongly, and because it would really mess up the selection steps that we're going to see going forward. And so this is sort of the view from your textbook, again, of showing that same process of testing where we first do the heavy chain rearrangement in our pro B cell. If it works, That means we qualify as a pre-B cell. Now we can make our different light chains. Once one of them works, we qualify as an immature B cell, and we are basically done with our VDJ recombination. So let's imagine that everything works perfectly here. At this point, our B cell is an immature B cell. Hooray! That immature B cell is going to go through this next testing phase that I'm going to tell you about. Let's just imagine it passes. And it will then actually get to leave the bone marrow and be a B cell that goes through the blood into the spleen and actually looks for microbes. You can see that in the bone marrow, it is largely making this black B cell receptor. And as it comes out, it actually starts making both a black one and the red one. That is IgM and IgD. If you recall, the M and D constant regions are the closest to the VDJ join. So when we get RNA made, we can get an RNA that includes both the M and the D constant region before the polymerase is like, no, I'm not doing anymore. What are you trying to make me do? I'm not going 55 kb and not doing anything. So we get this RNA made where we have the VDJ join, the M and D constant region information, and the cell can actually splice to make either an IgM transcript or an IgD transcript from that same DNA. Because this is an event with RNA splicing, it's reversible. And so the cell is going to start making IgM and IgD. Here you can see we have our cells that have been in the bone marrow. They have correctly made a B cell receptor. You can see their nice correct B cell receptors here. And they sort of leave the bone marrow, making both IgM and IgD. And their goal is to find a microbe like this virus and do stuff like make antibodies and all the things that they can do to protect you from different microbes. But as I said, there is this one more testing step that has to happen with this immature B cell. And it is related to this view of what we see, what I showed you for clonal selection earlier. So remember that when we're going through all of these recombination steps, we're doing that without looking at antigen. We're not trying to match an antigen. We're just randomly picking V's, D's, and J's and putting them together in the hopes that we're going to make something useful. And so we make this big diversity, and then when those cells finally leave the bone marrow, then we'll see if the thing they made was useful, if it binds to a microbe that we ever encounter in our life. And you can see that on the right as well. But there's actually a little additional piece to all of this that on this version I have covered up with boxes so you can't see. But now I'm going to have to show you the real version. When we go through the process of making this huge diversity of B cells with different B cell receptors, some of them respond to foreign bacteria. Yay. Some of them respond to, I don't know, some microbe that you're never going to be infected with ever in your life, and so they're there and they're useful, but you're never going to use them. But some of them bind to self-antigens. Some of them bind to proteins, lipids, nucleic acids, carbohydrates that are found in your body. Because we're doing this process of diversification at random, just making receptors that bind to basically any set of amino acids. And sometimes when you do that random process of making these receptors, the result is a receptor that binds to self things. That would be really bad. If you actually let those B cells leave the bone marrow and try to combat their antigen, make antibodies, the result would be you would have an autoimmune disease. The result might be if, for example, you have a particular type of anti-DNA antibody, that you'd have lupus, or a variety of other autoimmune diseases. This is the risk that the adaptive immune system has sort of evolved to have us take. We have evolved this process that allows us to make an incredibly broad repertoire. We make receptors against everything, including things we've never seen before, so that microbes can't be tricky and mutate and get around us. We already made every possible receptor there is. But in making every possible receptor there is, we went so broad that we actually made some receptors against ourselves. This is a problem that we see in B cells. This is a problem that we see in T cells. Your immune system basically has evolved so that you're right on the edge of autoimmunity. And a lot of places in development of B cells and T cells, if they get messed up, can push you over into autoimmunity. And so what we actually do is we take our immature B cells and we put them through a testing phase where we test them to see if they bind to self-antigens. If an immature B cell binds to self-antigens in the bone marrow, that cell gets taken care of. We make sure that that cell is not going to go into the rest of the body to lead to autoimmunity. So here you can see we made this big diversity of potential cells, and then we test them, and we only let the ones that did not bind self out of the bone marrow, where they can further look for antigens. And so this is sort of actually what the clonal selection theory states, where we have this version of getting rid of the self-reactive cells as well. One thing I will mention, I'm going to mention this now. It is going to become a thing that we care about later when we think about autoimmunity, and we're going to care about it a bit when we think about T cell development. Right now, you're kind of going to be like, why is she telling us this? It's one of those compare and contrast things. One thing that you should realize is that when we are testing our immature B cells to see if they are self-reactive, the only things that are available for the testing, the only antigens that are available for the testing are proteins, carbohydrates, nucleic acids, lipids that are in the bone marrow. So if there's some protein that's only found in the brain, we can't test our B cells against that. That's not available for testing purposes in the bone marrow. What I hope that gives you a hint on is we got to have a different way to deal with that. And that's again a later problem. But we really are only looking at things that are in the bone marrow. But pretty much if it's in the bone marrow, it's probably like everywhere in the body. And if you made a response to something that was everywhere in the body, that would be a catastrophe. So this is sort of the general idea of this process. When the way that we deal with these cells that are potentially able to interact with self, the way that we deal with self-reactive cells, is known as tolerance in immunology. So ways we deal with self-reactive cells are tolerance mechanisms. Specifically, right now, we are talking about some central tolerance mechanisms. Central tolerance mechanisms mean tolerance mechanisms that happen in a primary lymphoid organ, or tolerance mechanisms that happen while the cell is developing. There are also tolerance mechanisms that happen with the cells later in the cell's life, and we're not dealing with that now. Right now, we're just looking at how we work with these B cells in central tolerance. All right, I'm going to do the rest of this in a slightly weird order. And that's because there are some experiments that I haven't really talked about right now with B cell development. What I'm going to do is give you the sort of big overview of the tolerance mechanisms. I'm going to skip talking about some experiments, so I'm going to be skipping over a few slides, because I want to make sure I give you the basics of the tolerance mechanisms. When I am done, I may come back to those experiments, depending on time, but I want to make sure we get through the tolerance mechanisms first. Okay? So if you recall, when I talked about this process of dealing with those self-reactive cells, I said we deal with them. And some of you guys snickered because you thought I was making like a euphemism. But the reason why I said that is because there are actually 3 different things that can happen to those self-reactive B cells. The goal is that we don't want to have the self-reactive B cell leave the bone marrow as a self-reactive B cell. And so there are 3 different options of what might happen to that self-reactive B cell. And so those are the 3 mechanisms of B cell central tolerance. So you can see this view from your textbook here. If our B cell in the bone marrow does not interact with the self-antigen, it leaves the bone marrow, expresses IgM and IgD, and we all live happily ever after. If, however, that B cell does interact with the self-antigen in the bone marrow, then it is retained in the bone marrow so that one of these 3 things can happen to it. The first thing that can happen is implied on some of the slides I just showed you. And this first thing that can happen is called deletion. If that cell reacts with self-antigen in the bone marrow, and this is usually something that when it's a really strong interaction, that cell dies. That cell undergoes apoptosis. So you say, you cell, you are a bad cell. You would give us autoimmunity if we let you leave. We're going to kill you instead. So that's deletion. That's pretty simple. Experimentally, deletion was actually pretty hard to kind of examine. But when people examined deletion, in the experiments that I am currently skipping over, they found that deletion wasn't the only option. They found these 2 other options as well. One of these options is called receptor editing. And in receptor editing, our self-reactive B cell can try to make a new receptor. So its first receptor was self-reactive. It could try again to make a different receptor that is not self-reactive. There is one thing that is really, really, really terrible about this image on the left. When a B cell does receptor editing, The B cell specifically changes its light chain. It does not change its heavy chain. In fact, we'll talk about that in a second. So what you can see is we had this autoreactive cell on the right. It has the black light chain and the black heavy chain. And when it undergoes receptor editing, it will make a different light chain, either maybe this blue one or this pink one. But it's still stuck with that same heavy chain. Of course, it's the combo of the light chain and the heavy chain that's important for which antigen you bind. So just by changing the light chain, you get to bind a different antigen. This is such a nice image, except that they changed both the heavy and the light chain in it. It makes me sad. And receptor editing is a pretty good option for autoreactive B cells. Because receptor editing can happen in a couple of different ways. On the problem set, as you look at different practice materials, you will notice that a lot of times I'm having you draw the DNA before and after. And one reason why I have you drawing the DNA before and after is because it helps you understand some of the details of receptor editing. So here is our B cell. It has 3 V's on its light chain, V1, V2, V3, 4 J's. And this B cell is going to choose V3 and J3, which you can see here. And you'll notice that J4 is still present, as are V1 and V2. Now this B cell, it makes a successful light chain, but that light chain is autoreactive. We get a signal in the bone marrow that we are autoreactive. And so this cell could undergo receptor editing. And because of the setup of the mini genes in the light chain, and because we left V1 and V2 and J4, we can just redo the rearrangement and pick ones that are on the outside. So we could pick J4 and V2 and get rid of this autoreactive business and make a new one. And so with the light chain, you actually have multiple tries where if you happen to have used more inside V's and J's, you could use the more outside ones to get rid of the other ones because you've left those outside V's and J's. If it just so happened when you were picking, you picked J4, well, you're not going to be able to receptor edit. But if you picked inside ones, you can. And so you can see this here from your textbook as well. Here is my autoreactive cell. It has the blue heavy chain. It has this kind of whitish light chain. And that came from using, I guess, V3 and J1. We don't really know if there were other ones in between, but we'll say it's V3 and J1. And this B cell can be like, okay, self-reactive, let's try again. And now it can use V2 and J3. So it ditches this VJ join that it made and uses these outside ones. So now it's got this yellow heavy chain, or yellow light chain. And maybe it tests that one again. If it tests that one again, maybe that one's still self-reactive. Well, it could do it again and use the even more outside V and the even more outside J, if there are any left. If there aren't, it's out of luck. And then it could test that one, and maybe finally this third one, this orange one, actually works. Or maybe it doesn't, and the cell ends up having to undergo apoptosis. But the cell gets multiple tries because of receptor editing. And receptor editing is even more fun because it also has 4 chromosomes to play with, the 2 kappas and the 2 lambdas. So B cells get a lot of tries to receptor edit and not have an autoreactive light chain. They can use multiple V's and J's on the kappa and lambda chromosomes to try to get a good light chain. Yep, Carney.
Why can't we do the receptor editing?
It's going to be the next slide. Do you have a question, Carmi?
I don't know. I didn't hear her question, but I don't know if it's the same one. I was going to ask, is it only from the inside
Well,
you said it's from the outside. What
so
needs to change?
so realize that, notice that, say, J1 and 2 are missing here. They got deleted when we made this first rearrangement. So they're deleted. We can't get them back. So those would have been more inside, and we can't use them because we deleted them. But these other ones on the outside are still there, and so we can still use those. So it's about which pieces of DNA on the outside didn't get deleted during our original recombination. Does that make sense? Okay, yep.
For the bottom one there, you wouldn't be able to do it again, right? Because there's no other J.
Exactly. So here we couldn't do it again because we don't have another J with an RSS. So yeah, we do have another V, but we don't have another J, so we're out of luck. Yeah.
So if you start from the outside, there's no way that
Exactly. So if luck would have it that you started with V1 and J4, you don't have another chance on this chromosome. But lucky for you, you got 4 chromosomes. You got the 2 kappas and the 2 lambdas. So you have tons of chances to receptor edit and make a light chain that works. So again, like I said, this is why we talk about, why I said we deal with the cell, not we necessarily kill the cell, because one really nice option for the cell is to receptor edit. So Carney asked a great question of why can't we receptor edit at the heavy chain? And I'm going to show you this image. You can see light chain rearrangement at the top, and you can see heavy chain rearrangement at the bottom for comparison. So let's think about why we cannot receptor edit at the heavy chain based on what you can see here. So in fact, this view of the light chain at the top is exactly the same one you saw in the receptor editing example here. But this is an example of a heavy chain. So there is a problem. Yep, Jameer.
All the D's are removed when you're doing VDJ.
Yeah, when you do heavy chain, you've got V, D, and J. V and J have the same kind of RSS. You have to have a D in the middle. When you put D and J together, you get rid of all the other Ds that are between D and J. And then when you put V and DJ together, you get rid of all the Ds. So in this situation, you might have extra Vs and extra Js, but you don't have any extra Ds because you deleted the Ds in these earlier steps. So it's not, and so you can't do V to J and skip D because your RSSs don't work. You've got the same RSS here. You needed the D to have the right RSS to join all that together. So because you actually get rid of all those Ds in making the heavy chain, no receptor editing possible at the heavy chain. But because the light chain just has V and J, and you end up with some of those outside gene segments still present, you can receptor edit at the light chain. Does that answer your question, Carney? Okay, cool. Other questions? All right. So now we've got option number 3 of what can happen to this autoreactive B cell. Option number 3 is the crazy one. So I want to think for a second before we get to option number 3 about some scenarios that we might have to think about your immune system having to deal with. So first of all, I want you to think about deletion and this idea of deleting our autoreactive cells. Anything that seems kind of sad about that, especially if you were like the ATP accountant? Yeah. Oh my gosh, you just put so much energy into that cell to do all of these steps, and then you just killed it. That kind of seems a little wasteful. Not gonna lie, the immune system, so wasteful. We waste things all the time in the immune system. But it's a little wasteful, right? In some ways you're like, man, I wish I could save this cell. Well, You might be able to. You might receptor edit. But what if when that cell is binding self-antigen, what if it binds it kind of like, not super strong, but definitely binds. Not nothing, but there's some binding. Now how do you feel about that cell? How do you, especially if you're the ATP accountant? Mine's a little bit to self. Now maybe you're like, boy, it's even harder to delete this cell. Because like if it was binding to self really strong, then it's going to give me some terrible autoimmune disease and I'm going to die. But maybe if it only binds a little, it's not like so dangerous. And maybe now I worry more about if I wasted that cell. And in fact, with these three options, the third option is the option that we tend to see for somewhat weaker, more weaker self-reactive cells. When we think about VDJ recombination and we think about making all of these receptors, we're making receptors that bind to biological molecules, right? That's the idea. That could be some random microbe that it could bind. Some random biological molecule. And we're also made of biological molecules, so that's where the risk is. Our biological molecules and the microbes aren't always super different. Sometimes they can be really similar. So now what if I tell you that that B cell that binds to self, kind of weakly, kind of meh, also binds to Ebola proteins perfectly. It is the best ever Ebola fighting B cell. Because it turns out one of the Ebola proteins and one of our proteins are similar enough that it could bind our protein weakly, the Ebola protein, perfectly. Now how do you feel about that B cell? And what do you want to do? You want to preserve it. Why do you want to preserve it? Maybe it's still useful, right? Maybe if you're in Uganda right now, where there is currently an Ebola outbreak, that B cell could be really useful. Maybe if you're me in Madison, New Jersey, that B cell's not that useful, and the risk is worse. So what you'd actually kind of like to do is maybe save that cell, preserve that cell, but somehow try to reduce its risk a little bit. Oftentimes, when you are in a very bad situation infection-wise, if you have Ebola, if you have COVID-19, this is a big part of what goes on with COVID. If you have a lot of different infectious situations, you make so many cytokines. You make all the cytokines. You have like crazy cytokines going on in your body. So what it turns out we do is we basically take that cell that's sort of weakly self-reactive in the bone marrow, and we turn it off. We let it leave the bone marrow, but we let it leave the bone marrow turned off. So that if it sees its antigen, it can't do anything. Now again, you're like, this seems like the dumbest thing ever. Now I'm wasting more ATP. But that cell is off unless it sees like the sledgehammer of cytokines. Unless it's a total emergency with all the cytokines. And then if there's a total cytokine emergency, that cell gets turned back on. Because if you're at the point of an emergency, you need all the help you can get. Does that concept make sense? Because oftentimes people hear about this idea and they're like, what happens to it? it's allowed to leave but is off mostly, but could, like, the idea is it leaves the bone marrow because it was weakly self-reactive, but is in this state of being off for all intents and purposes unless craziness happens. And so you can imagine that cell as being off. This option is known as anergy. And so if we have kind of weaker binding to self-antigen, that cell is going to get a block, so it cannot signal anymore, but it's going to be allowed to leave as an anergic B cell. It's going to be hanging out in your body off. And you can see this version from your textbook as well. Here's the cell. It gets a low affinity self-antigen. It's allowed to leave the bone marrow, but it leaves the bone marrow and becomes unresponsive. And then everyone asks the question of what the heck is the point of it? Well, the point is that if there was a crazy emergency, it could respond again. But under all normal circumstances, it is unresponsive. In the experiments that people use to talk about these cells, one of the things that Siri really wants to be involved in, one of the things that we often talk about, which I feel like mid-semester students are starting to understand this concept pretty well, is that these cells are present but not functional. And I can tell you there are definitely meetings I have been to in my life where I have been present, but I have not been functional. And so these cells are there. You can measure them. You can find them in the body. But if you actually try to stimulate them and try to make them do things, they don't do anything. So that's what this anergic cell is, or this energized cell is. Yep?
So is the cell still transcribing and making reciprocal dividing?
It will probably be doing those things at a very low level, unless it gets sledgehammer signal, because it's the emergency and we need it at some point. Okay, so those are our three options. Once our cell is able to leave the bone marrow, it goes into what immunologists refer to as the periphery. And the periphery is basically everything that's not the developmental organs, everything that's not primary lymphoid organs. Usually, those cells are going to go to secondary lymphoid organs. Those cells are going to do things like make lots of antibodies to protect you, make memory cells to protect you for a long period of time. All that good stuff is secondary lymphoid organs in the periphery. One super important thing is that at this point of the B cell's life, it stops making RAG1 and RAG2 and will never make it ever again. When that B cell leaves the bone marrow, it is done with RAG1 and RAG2. Because it does not want to have that dangerous cutting, pasting enzyme present. So we are going to stop making RAG1 and RAG2. No matter what, we are never going to make it ever again to avoid DNA damage. And now we're going to do things like search for microbes, find microbes, attack microbes in the pink organs. In order to make this work in most settings, our B cell needs to interact with a T cell. So in the periphery, B cells often need to interact with a T cell through the mechanisms shown here. As a result, I cannot tell you anything else about the life of the B cell until after we learn about T cells. And that's why, once the exam is over, we start T cells. And it's going to be kind of the same thing we did with B cells, where first we're going to spend some time thinking about the antigen, and then we're going to think about the receptor and what the cell does. So it's going to be a very parallel situation as we talk about T cells. One piece I will just point out is that I have been talking about B cells as if there's just one kind of B cell, but there are really tons of types of B cells. In fact, whenever I talk about this, my little sister, my PhD was all about T cell stuff, and now I do innate immunity research. And my little sister's PhD was all about B cell stuff, and she always like gets on my case for like underestimating B cells and not like talking about all the kinds, because there's so many kinds, and how dare I not talk about all the kinds? So there's lots of kinds. You could ask my sister about them if you want to know about them. But I will just say that in particular, when I'm talking about B cell responses, I'm largely talking about what are known as B2 B cell responses. There are others, including this one type called a B1 cell that's found in the peritoneal cavity, and it is actually a little bit, has some innate-like functions. And so we are going to talk about kind of innate-like functions and some such cells a little bit later in the semester. So I'm going to spend the rest of the time today talking about some experimental stuff. When we think about B cell development, we often will look at measurements of B cell development using flow cytometry. And so we should think about how we would use flow cytometry to imagine some of the different types of cells. So I'm going to give you a couple of examples. Basically, I'm giving you, it's gonna feel like the two examples are really similar, because they are. They're just slight twists on each other. So I'm gonna tell you that let's imagine that I took some bone marrow and I did some flow cytometry on the B cells in that bone marrow. So I have already done some kind of gating or some other kind of thing. So the only thing in my little tube is B cells. But it's B cells that might be pro-B cells, or might be pre-B cells, or might be immature B cells, because I'm looking at the bone marrow. I can distinguish them by flow cytometry pretty easily. So the big way that I might do this is I'll do my flow cytometry plot, and the two antibodies that I will look at will be an antibody that binds heavy chain. Usually we say this is an anti-mu antibody, because it's binding the IgM part of the heavy chain. We could also have an antibody against light chain. Right now, I'm just going to say it binds light chain. We're going to complicate that in a moment. And I can have my four little areas, my four little quadrants. If I did this plot, where would I find pro-B cells? Jameer. In the bottom left quadrant. Why would I find pro-B cells there? Right. So our pro-B cell has no heavy chain and it has no light chain. That's the definition of the pro-B cell. So it's going to be negative for heavy chain. It's going to be negative for light chain. So the cells that fall into this region are my pro-B cells. Okay. Where am I going to find my pre-B cells? Michael. Bottom right. And why am I going to find my pre-B cells here? So these cells have made heavy chain, but they have not yet made light chain. So I can find them right here. Then where am I going to find my immature B cells? Michael? Top right. And why am I going to find the top right? Because these cells are defined by having heavy chain and light chain. Should there be any cells up here in the top left? So Jameer says no. Why no? Yeah, there's no this would mean a cell with a light chain and no heavy chain. And that's not a thing. So nothing up there. So this is kind of the simple version of what you might see here. In reality, it's actually slightly more complicated. And the reason why it's slightly more complicated is because when we use our antibody that binds to light chain, we have to pick an antibody that binds to either the kappa or the lambda light chain. So antibody that binds, we'll say specifically kappa now. If I actually specify that we're talking about kappa light chain, pro-B cells don't change because they didn't have a light chain in the first place, right? Pre-B cells don't change either because, again, they only have a heavy chain. We still haven't done anything with the light chain. But the immature B cells will be a bit different. This is actually going to be the immature B cells that used kappa, the ones that chose kappa. But remember, there are also some immature B cells who chose lambda. Where are the immature B cells who chose lambda going to be? Yeah, Jameer? Top left quadrant, so they have light chain and no heavy chain? They have kappa light chain and no heavy chain? No. Where are they going to be? Yep, Carney. Bottom right corner. So this is actually going to be the pre-B cells and the immature B cells that used lambda. And I might have to do something else to distinguish between them. Yeah. So here it does not come up, but I have seen situations where you could think about what would happen if you had heavy chain and you had the fake light chain lambda 5. Then where would the pro-B cells be? So they'd still be here. Where would the pre-B cells be? Top right. They have heavy chain and they have fake light chain or surrogate light chain. And then where would the immature B cells be? Yeah. They have heavy chain, but they don't have surrogate light chain. And so you could distinguish between all of these different types of cells with these types of things. The other way you could look at this is sometimes I've asked the question. Here, I'll make up a question that looks like a question I've asked on past exams. You can tell me the answer. Actually, I'm going to do it this way. Okay, so I got 3 plots. Okay? Here's my story. I did an experiment where I got some B cells in the bone marrow, and I sorted them into pro, pre, or immature B cells. So I had 3 tubes, pro, pre, immature B cells. And I was going to run them on the flow cytometer. And I let my lab assistant help me. And we usually make the lab assistant from FDU. because when I went to college at Duke, we had the rival of UNC, and so we always made it from UNC. So I tried to figure out who would be the. So your lab assistant from FDU comes and does the experiment for you. And they forget to label which one was pro-B cells, pre-B cells, and immature B cells. And this is the data you get. So tube 1, tube 2, tube 3. Which one was pro, which one was pre, which one was immature? Yes, Ermi. You can say all of them, or you can just start with one. Pro would be number 3. We're negative for heavy chain, we're negative for light chain. So that must be the pro B cells. Okay, which ones are pre B cells? Yeah, Jay. The first one. Why the first one? Mm-hmm. So note that this is lambda, not lambda 5. Remember, the light chain is kappa or lambda. The surrogate light chain is lambda 5. I made it lambda so we could have that discussion. So this is the, just like you're over here, light chain was kappa, now light chain's lambda. So which one is pre? Yeah, Jameer. Yeah, so this is pre. We've got heavy chain and no light chain. And what's this? Yeah, Michael. Immature, and how do you know that?
Because the receptors that have the lambda light chain and the heavy chain top rate have kappa light chain on the bottom.
Yeah, so heavy chain kappa light chain, heavy chain lambda light chain. So these are the immatures, these are the pros, these are the pre's. So you can see how we can use the flow cytometry that we've been doing all semester, as well as kind of these definitions of these different cell types together to actually think about what an experiment would look like. I have 2 minutes left, and I'm going to give you those 2 minutes. So you will be getting lots of emails from me, and the review session, if you are interested in it, will be on Sunday at 7 on Zoom. My cat will be very excited to make an appearance. And I will see you guys on Monday, if not online before.